Okay, so we are going to go in and finish chapter 9. So, just as kind of a nice reminder, um, what is enzyme activity controlled by? And we've talked about this multiple times. We said, right, substrate availability is one way that we can control how quickly a reaction happens, right? But if we want to actually change enzymatic activity, we have to alter the catalytic activity, right? But we can control which reactions are happening in the body through substrate availability and through changing catalytic activity. So the same two big components are going to control flux through every different pathway. And so metabolic pathways, this example of glycolysis is one of them. So we know that there are quite a few reactions in glycolysis that have delta G values that are close to zero, right? Plus or minus a little bit, but basically zero. And those are all reversible. So when you look at this pathway, as we're going down this way, we have glycolysis. As we're going up this way, we have gluconeogenesis. So if you're going down the glycolysis pathway, the reactions that are shown are those that have very high negative delta G values. The other ones just kind of aren't shown here. So we know that basically the direction of flux of our metabolic pathway is really a lot dependent upon substrate availability, right? So when are we going to make pyruvate, right? When are we going to go through glycolysis and break down glucose to make pyruvate? When we eat a lot of sugar, okay. But we also have rate limiting reactions, and those are the ones where we can actually decrease enzyme activity, even though we have lots of substrate sitting around. So the reactions that have those high negative delta G values are really functionally irreversible because of our physiologic metabolite concentrations, and they're subject to enzymatic control, right? So which three enzymes fit this definition? And those are hexakinase, phosphofructokinase 1, and pyruvate kinase. These are called the rate limiting steps in glycolysis, because you could only have enzyme activity, right? Well, let me put it this way, you can change the enzyme activity without worrying about what the substrate concentrations are, right? So I can have inhibitors and activators that are independent of substrate levels. So the reverse of glycolysis, we've talked about glycolysis over and over and over again, but what we really haven't talked about too much is gluconeogenesis. So gluconeogenesis, right, is that, and it's not really the reverse, but it's sort of the reverse of glycolysis. It requires ATP, it's anabolic, whereas glycolysis produces ATP and it's catabolic. So it uses the same enzymes for all of those reversible steps. So all of the parts in the pathway that are here, these are all reversible portions of the pathway. The parts that aren't reversible are the things that have very high negative delta G values. So what we're going to talk about is how we can bypass that reaction. So if we have to go in reverse, right, we're going to start at the bottom, we'll start at pyruvate. And instead of using pyruvate kinase, right, we're going to use two different enzymes. So we need two enzymes to bypass one. So our gluconeogenic enzymes are pyruvate carboxylase and phosphoenol pyruvate carboxykinase. So those two enzymes do the job of pyruvate kinase in reverse. So that's one bypass area, right? And we need negative delta G values for that, and we do. Then the next step, if we're going this direction in gluconeogenesis, is we need to bypass the PFK1 reaction. The PFK1 reaction is bypassed by fructose 1,6-bisphosphatase. So this one, it's a 1-to-1 basically, and you can look and they have very similar negative delta G values. And then that very first reaction in glycolysis, the last one in gluconeogenesis, we're going to substitute hexakinase for glucose 6-phosphatase. Remember, these phosphatases, these are the ones that pull off the phosphate groups that the kinases put on, right? So we kind of have an understanding that the same things happen in reverse, but we just use different enzymes in order to flux between glycolysis and gluconeogenesis. So if we look at some of these steps, like these regulatory steps. So we said in glycolysis, that's phosphofructokinase 1, and that's how that one is regulated. So we talked about the energy charge of the cell, ATP, AMP, and ADP being regulators of PFK1. And so these are all what we call allosteric regulators. They bind to the enzyme, they induce a conformational change in the protein. And so it changes the conformation of the active site. And so that alters the activity of PFK1. So PFK1 is kind of like hemoglobin, where you have that T state and R state. So for PFK1, our T state is where we have ATP. So if you have, remember your 10th state, we didn't like, I guess I should do that in red, right? So it would be the T state. Whereas if we had ADP or AMP, we would be in the R state, right? And so it's just talking about activity of the enzyme. So we talked about those. Citrate. Citrate is another inhibitor of PFK1. So you got to stop and think about this one. Citrate is made in the citrate cycle. It's actually the first product of the citrate cycle, which is why I like calling it the citrate cycle and not TCA and all that kind of other stuff. So if the citrate cycle is backlogged, is working and it can't keep up, right? So then the citrate cycle is basically going to stop. So citrate's going to accumulate. Then citrate is going to come over to the glycolytic pathway and it's going to inhibit phosphofructokinase 1. And that all makes sense, right? Because basically citrate cycle is going to shut down when there's tons of ATP. Well, the same thing is if I'm going to shut down citrate, I also want to shut down glycolysis because I don't need any more ATP. And then the last one that's an allosteric regulator is the fructose 2,6-bis phosphate. And so this one is a metabolite that controls carb metabolism. And if you take biochem 2, you'll find out about it then, but I'm not going to talk about it anymore in this class. All right, so remember last lecture, we were talking about how certain cells have certain enzymes that phosphorylate glucose, and that glucokinase is only in liver and pancreatic cells. And so the liver cells, we talked about making glycogen, but let's look at the pancreas. The pancreas is where we are going to make insulin. So how does that work? Remember that glucokinase is that glucose sensor, right? It's the one that is going to have a low affinity for glucose. So it's only going to really respond if there's really, really high blood sugar levels or high blood glucose. So it's going to function as that sensor by inducing insulin release only when you have high blood sugar levels, right? It does the same job as hexokinase. The difference is the affinity and that it's not inhibited by the product, which is really kind of important. So if we look, right, glucose comes in to our cells, our pancreatic cells, gets transported in through GLU2, and that glucose goes through gluconeogenesis. But the gluconeogenesis is dependent on glucokinase, not hexokinase. So basically, no matter how much you go through the glycolytic pathway, it's not going to inhibit. So as long as your blood sugar is high, your pancreatic cells will be doing this process, which is good, right? As soon as they stop, then you have a problem and you have diabetes, right? So we're going to go through glycolysis. We are going to make pyruvate. Our pyruvate is going to be converted eventually to ATP, just like it is in all the normal cells. But what's different about pancreatic cells is once you have a high level of ATP, it's going to inhibit potassium channels that are on the membrane of the cells, the pancreatic beta cells. And so that's going to induce depolarization of those membranes. When that depolarization happens, it has effect on a second transport protein, and that is a calcium voltage-gated channel. So voltage gated means if the membrane's polarized versus depolarized, it'll be open versus closed, right? So it's depolarized, so it's going to be open. Calcium is going to rush into the cell. That locally high concentration of calcium in the cytoplasm is going to stimulate these vesicles. Now, these vesicles are filled with the hormone insulin. And if you have those high levels of calcium, which means basically that you had high blood glucose levels, then these vesicles are going to merge with the membrane and the insulin will get pumped out into the bloodstream. And that's exactly what you want, right? You want to have insulin so that that insulin goes to your other cells of the body and tells them, hey, you have high blood sugar, you can take some glucose into your cells, which is awesome. All right, so where do we get glucose for glycolysis, right? So the answer is actually a lot of different places, right? So our liver cells, our muscle cells, we're going to obtain glucose from our diet, and we can get it from glycogen, but we're going to kind of primarily talk about the diet here. So if you look in the middle, right, if we look in the middle here, in this blue box here, this blue box here, what we can see is this is the glycolysis pathway. And anything that is boxed in blue is a glycolytic metabolite. So it is a substance that is in the glycolysis pathway. And those red arrows that are coming in, those are where our different metabolites are actually entering glycolysis. And we like to think about glycolysis as a linear pathway, but we don't always enter as glucose. We can. So if we eat things like sucrose, sucrose is a table sugar, right? And it's actually a disaccharide. And so that's why it's boxed in yellowish, right? Because it's a metabolite that can be broken down. So when sucrose, the disaccharide, is broken into monosaccharides, it will turn into fructose and it will turn into glucose. And so the glucose can go in and directly enter the glycolytic pathway, but fructose cannot. So fructose would have to be phosphorylated to enter, wow, that's crazy, to enter as fructose 6 phosphate. I don't even know how all that happened. Okay, anyway. So there are different places where things that we eat can enter the pathway, right? The fructose can then go on and be converted to fructose 1 phosphate. And fructose 1 phosphate can be converted to glyceraldehyde, and then glyceraldehyde can be phosphorylated, and you can make glyceraldehyde 3 phosphate. So you could enter it at a different place. So there are lots of different ways that you can have things come in. Maltose is an example of another disaccharide that is 2 monosaccharides of glucose hooked together. So if you take maltose and you break maltose down, you'll get 2 glucose. So that can come in the beginning. Lactose, on the other hand, the one in milk, right? And maltose is a fruit sugar, right? It's found in fruit. But lactose is in our milk, and lactose is 2 monosaccharides hooked together to make a disaccharide. And so you have one that's glucose that comes in directly. I don't know how in the world I'm doing that. Okay. It could come in directly as glucose, or it could turn into galactose. Either way. But if you have galactose, galactose cannot immediately enter the glycolytic pathway. It's not a metabolite. So you would have to convert that galactose to glucose. And there's a whole pathway that you have to go through to make that happen. But most people who have lactose intolerance, their problem is that they're missing enough of the lactase gene that splits the disaccharide into monosaccharides. Okay. The other thing that can happen is, right, if you don't eat sugar, you have fats, right? So we can go to our fats for energy. So how does that happen? Well, you have 3 fatty acid tails and a glycerol molecule that make up a triglyceride. So if you separate the fatty acid tails from the glycerol, the fatty acid tails are then metabolized into acetyl-CoA, which can go into the citrate cycle. The glycerol gets converted eventually to dihydroxyacetone phosphate, which is a substrate in the glycolytic pathway, right? So you can get energy even from the glycerol backbone. I thought that's kind of cool. All right, so let's take a quick look at the galactose metabolism pathway. So remember, our lactose is composed of galactose and glucose. The glucose goes straight into glycolysis, but galactose cannot. So there are 4 different steps that have to happen in order for galactose to enter the glycolytic pathway. So you first have an enzyme that phosphorylates it. So we go from galactose to galactose 1-phosphate. Now, even though we are phosphorylated, we cannot use that in glycolysis. Our enzyme structure does not fit galactose. So basically what we do is we exchange it. We exchange galactose 1-phosphate for, you have this molecule called UDP glucose. UDP glucose is just like ATP, GDP, CDP, right? All of those different nucleotides, it's the same thing. So this one just has the sugar glucose attached to it. So we swap the glucose for the galactose. And when we do that swap, we get a free glucose 1-phosphate, and we make a UDP galactose molecule, which can then undergo an epimerization reaction where we regenerate the UDP galactose. So it can be used again in another cycle and another cycle. And then the glucose 1-phosphate is again not quite right for glycolysis. So we actually have to undergo a mutase reaction where we move that phosphate over from being over here on carbon 1 to being over on carbon 6. And then we're ready for glycolysis, right? So we can keep going. So we get things into glycolysis a lot of different ways. And then all those substrates inside of glycolysis can actually leave the glycolytic pathway before the pathway is complete. So the glycolytic intermediates are really important in lots of different pathways where we have to make things. So anabolic pathways. So if you just look at our first substance, look at glucose. The red arrow means that glucose can go out to make different things. So we can use glucose, we can go through the pentose phosphate pathway, and we can make nucleotides, right? We can also use those pentose phosphates to actually generate energy carrying molecules like NADPH in order to use them for reducing things, for all kinds of different purposes that you'll learn next semester. Please take biology, biochem 2. All right. So glucose could also go, if you have a ton of excess of it, right? If we have phosphorylated on that first carbon, then we can go and store it as glycogen. That's always possible. We could use fructose to go and make glycolipids and glycoproteins, right? So that's something that's really, really important for cell signaling and all that kind of stuff. We said, just like we said, you can use the glycerol from a triglyceride to make dihydroxyacetone phosphate. We can also use dihydroxyacetone phosphate to make the glycerol to make triglycerides if we have very high energy charge in the cell. So a lot of things, just like glycolysis and gluconeogenesis, are reversible. And remember, we talked about this, our 1,3-bis phosphoglycerate being able to turn into 2,3-bis phosphoglycerate to regulate oxygen transport. We talked about that. And then a lot of these different glycolytic intermediates can turn into all of these different amino acids. So there are lots of different ways that glycolytic intermediates can provide the carbon skeletons for the biosynthesis of all kinds of things. So super, super important. All right, so when we finish glycolysis, what do we have? We have 2 molecules of pyruvate. So what's going to happen to that pyruvate? Well, it depends. It depends on the organism, and it depends on how much oxygen is available. If there is no oxygen available, right, we're going to go through anaerobic. If we have oxygen, we're going to go through aerobic. So the aerobic conditions, we go to the mitochondria, and inside the mitochondria, we go through the citrate cycle, we go through electron transport chain, we have oxidative phosphorylation, and we make ATP. And we also make, right, CO2 and water, but those are really waste. Under anaerobic conditions, so for example, this would be in muscle cells. and you're doing some weightlifting, you're doing some really strenuous activity, and you're going to take pyruvate and you're going to convert that pyruvate to lactate via lactate dehydrogenase, right? And it's really reversible, but we're going to use up NADH and we're going to make NAD+. Now, why is this important? This is so important because we need more NADH for glycolysis. If we never regenerate that NAD+, we can never go through glycolysis, and that becomes a huge problem. In different other organisms, not us, right? Saccharomyces cerevisiae, we can convert pyruvate, pyruvate to CO2 and ethanol, right? We can use it to make ethanol. But you also see it in other microorganisms, right? The bacteria that are used to produce yogurt, those that are used to produce cheese, things like that. So if we look at kind of a schematic diagram of how pyruvate can be metabolized, if you look this pathway here, this is our pathway in the muscles, right? And what you're going to notice, the second pathway that we talked about, going from pyruvate to ethanol, and then we talked about pyruvate going through the electron transport chain and making carbon dioxide and water as waste products. But what you see about all three of those different fates of pyruvate is that you are going to be making, so if you come out this way, you're going to be making NAD+. You make NAD+, and here you're going to be making NAD+. You initially make NADH, you finish going through electron transport chain, you make NAD+. So it's a little complicated when you get to the mitochondria. I'm going to show you very briefly why, but if you take biochem too, we'll talk about it a lot more. But if pyruvate goes to the mitochondrial matrix, right, it'll get fully oxidized and you'll end up with just carbon dioxide and water. And you're going to regenerate that NAD+. But the problem is that the NAD+ and that is made over here, the NAD+ that's made inside the mitochondria is stuck inside the mitochondria. And so you can't actually get it out. And so what you have to do is you have to have a shuttle system. And so the shuttle system is embedded in the membrane of the mitochondria, and it will allow you to exchange mitochondrial NAD+ for cytosolic NAD+. Then that NAD+ can go through glycolysis. And why do we need NAD+ for glycolysis? That's our glyceraldehyde 3-phosphate dehydrogenase. So the GAPDH enzyme is the one that requires NAD+ in order to proceed, right? So it's really, really, really critical that we regenerate that oxidized form of the NAD+ coenzyme. Otherwise, it's done. It's cooked. You're not doing it. You're not, the more you exercise, you're just going to actually damage your muscles. So we see this in people who have lactate dehydrogenase deficiencies. So if you have somebody who has a mutation where you have a loss of function of lactate dehydrogenase, then the patients can't maintain really long sustained periods of lots of exercise because they can't use glucose to make ATP that's needed for muscle contraction. They'd have to resort to like fats and stuff. But under anaerobic conditions, right, you have to have something that's going to convert that pyruvate to lactate. And when you don't, you're going to get muscle fatigue. You can even get muscle damage as cells really run out of ATP and become critical. It's really kind of dangerous. Okay, but on a lighter note, on a more fun note, right, we can use that lactate to make alcohol, which is interesting. So brewer's yeast is Saccharomyces cerevisiae, and we have been doing this for hundreds of years to make beer because we know that under anaerobic conditions, the yeast are going to rely on glycolysis to make ATP. So in the process of doing that, they make pyruvate. And the pyruvate can be metabolized by alcoholic fermentation to regenerate the NAD+. And we're going to capitalize on this. So what they do is they actually take some source of carb in here, and it's usually like a cereal grain, like a barley or something like that, and they germinate it. And so what does germination do? Germination induces expression of amylase, of maltase, right? So these are all enzymes that break down sugars. So then they get a mash, so they like mash it up. And so it releases the enzymes from the cells. You add water and you get what's called the wort. And so this is where you increase the temperature, you activate the enzymes that that were released from the cells. They're going to degrade the carbs, and they're going to make metabolites for the glycolytic pathway. So they remove all the cell debris, the undigested cell debris. The wort is boiled with something like hops, right? If you like a hoppy beer, this is where they put it in. And then they cool it. And after they cool the wort enough, they add yeast. You can't add it when it's hot or you're going to kill the yeast. So they add live yeast cultures after it's been cooled, but under aerobic conditions to promote growth, right, of that starter culture. But they only let it happen for a short amount of time. So they pretty quickly switch it over to anaerobic conditions. And then you have fermentation of the remaining sugars, and you're going to produce alcohol, right? So you produce ethanol and carbon dioxide. And so that's where you bottle it. And so really big companies will actually have the beer aged, they'll filter it, and then they'll recarbonate it. But smaller beer brewing places will usually do a second fermentation right at bottling to carbonate the filtered beer. So you get like fresh sugar and fresh yeast, and then you get that alcohol content, but you also get that carbonation. So it just depends on how you carbonate it, right? Whether you're a big company or small company. But all really important things for glycolysis. All right, that is it. Y'all have a great rest of your day.